My name is Neha, and on behalf of the Murty Trust, I would like to welcome you all today for coming here, for being with us, for attending, for braving the traffic and the rain. Given the circumstances, we'll wait just a few minutes, 4 or 5 minutes, for others to make it as well through the rain and through the traffic. So thank you for your patience. Just a few minutes more. I'm excited to be a part of this unique and powerful experience that we will be in together today. Briefly, here is our program. After a few quick notes from some very popular people in the audience, Manu will begin his talk at about 5:15 p.m. After the talk, we'll have a Q&A session followed by refreshments outside. Please scan the QR code that I would like projected up here from whoever is moving the slides. You would also have seen the QR code outside in a few places. You scan the code and you can submit your questions, and those are the questions that will be brought up in the Q&A session. Once you have done this, I request that as a sign of respect to your fellow audience members, please switch off your cell phones for the next 60 minutes so we can be truly immersed in the world of science. Without further ado, please welcome on stage our very own Mrs. Sudha Murty, author, educator, philanthropist, Murty Trust chairperson, and someone whom I, like many of us, very dearly look up to.
Hiriyarige namaskara. It's good evening to everyone. Today you have come to listen to Manu Prakash, not me. I'm aware of it. From time immemorial, India had a scientific bent of mind, and you can see, BC AD of Indian history. We have Varahamihira, Madhava School of Mathematics, Brahmadatta, and many people. But this is the tradition did not continue later due to various reasons, and now we want, in a new India, we would like that should be a lot more scientific approach in the society, because if science doesn't progress, then country will not progress, society will not progress. I remember when I joined engineering college in 1968, everybody asked me, because there were no girls in engineering college, they asked me, why do you want science? Girls, there's unthinkable a girl will do a science in those days, particularly engineering. And this is a quote I learned somewhere, that if human beings have to be happy and get life should be simpler, then you require applied science. To have applied science, you should have science first. So I request all of you, particularly young girls from Mysore, Surguru, you have come. And in that, girls, I am telling you, scientific bent of mind is most important thing in real life to progress, to understand, and can remove all your blind beliefs, and country is progress depends on that. In view of that, a friend of mine, by the way, by the name Arvind Gupta, you must have heard, Khel Khel Mein has written a book with the simple things. He has created a lot more scientific toys, like how Manu Prakash will tell you now with the paper he can do a microscope. You require a creative mind, you require a questioning mind. You should not accept with a blind belief everything. You should question why and make science easy, accessible, so that we can live a better life. On behalf of Murty Trust, I welcome all of you. People have worked very hard. It is not a vote of thanks, but I should not forget. Neha, Manal and their team have worked very hard, and please enjoy. And this is second in the series. Last year Dr. Srinivas Kulkarni gave the first one, and we would like to do many more such lectures so that people like Manu Prakash, who have done so much good work somewhere else, they should be introduced to our young audience. Thank you. Jai Hind.
Thank you, Mrs. Murthy, for leading us into the talk with inspiration. Now, I am delighted to introduce Professor L.S. Shashidhara, Director, National Center for Biological Sciences. He is a force for science, a change maker, and we are lucky to have him as one of the most prominent scientific leaders in our country.
Ellargu namaskara. This is the first time actually I'm here in this famous hall. I had read so much about Puttanna Chetty public lecture hall. It is actually built by Diwan at that time, Sir Mirza Ismail, with the help of Puttanna Chetty's contribution, both the land and some money he contributed, a philanthropist. And it has actually witnessed in the last 90 years several fantastic events, and also of course public talks, and today is one of them by Manu Prakash. We are happy to collaborate with Murty Trust to organize this public lecture. I represent here National Center for Biological Sciences, Bharatiya Jeeva Vigyana, Rathva Rashtriya Jeeva Vigyana Kendra antivi Kannadadalli. As the name indicates, we are a research institution in the area of biological sciences, and not any one particular area of biological sciences, across the biological sciences. When you say across biological sciences, it starting from a single molecule, it could be protein or nucleic acid, and then at a cellular level, like a bacteria or even a human cell, individual cell. We are made up of 50 trillion cells, but every individual cells can be studied separately. Or at the whole organism level, like a whole human being or an insect or a fish or a plant, and also population level. As you know, there is a huge difference between how you study an individual versus how you study a population. And then we can also study ecological level, where ecological multiple species interact with each other. At the population within the same species, many individuals interact. An ecological one, or more like a forest is an ecological one, or a human, bacteria, virus, this interaction is ecological interaction. For example, COVID pandemic is actually an ecological problem, although we consider it as a major disease in our context. So NCBS also wants to promote as much as possible the modern science for the benefit of the society. There are two ways of doing it. Whatever the science or the new knowledge that is produced is taken to the public and through an outreach. Second is our own research can be converted into a translational product for the purpose of biomedical or for agriculture related benefits. So through this, NCBS is contributing to the nation for the last 30 years. NCBS itself is a center of TIFR, which is much older. It is set up by Homi Bhabha in 1946 with the help of Jamshedji Tata, JRD Tata. And later, Indian government took over and made it an autonomous center within the Atomic Energy Commission, Department of Government of India. With this short introduction about NCBS, just about today's talk, Manu Prakash, he did his early studies at IIT Kanpur. He is a BTech in computer science. And as you know, computer science actually started in IIT Kanpur. It is the second in the world to start a BTech engineering program after Purdue University. And one of the best schools, and is also very, very competitive. I'm sure all of you know how competitive it is to get into a computer science program, particularly in IIT Kanpur in those days, and even now. And Manu, was trained in that one. But he moved very quickly to science because of his passion for asking questions and discovering new knowledge. As you all know, the process of science involves asking a question, making a hypothesis based on the prior knowledge, and validating it. When you say validation, it is not providing proof. It is finding evidence to prove or disprove. Most likely, in the way scientists we do it, it is by disproving our own hypothesis. And if we cannot find evidence, sufficient evidence to disprove ourselves, we assume that perhaps our hypothesis may be correct. This process is reiterated multiple times with the help of community. We get it peer reviewed, and at the end of it, it becomes knowledge. An opinion or an hypothesis or even an observation may become knowledge only after it goes through this rigorous process. Now, to do this kind of research, obviously you need tools. Why do we need tools? Because our sensory perceptions are very limited. Whatever we see, what we hear, what we can smell, what we can taste, or what you can touch and get a feel, it is not enough to understand the outside world. That's because we are all product of evolution and certain kinds of sensory abilities we have, but that's not enough to understand the whole world. For example, bats can see with the help of ultrasound waves. That ability is not there in humans. Butterflies can use UV light to see the world. That ability we don't have. But we have certain abilities which other animals may not have. For example, our language skills and other tool using abilities and so forth. So that means we had to, if you want to understand the world, you need certain kinds of tools which will help us to understand the world beyond our own sensory perception. Right? What Galileo used like a telescope, many people use microscope and so forth. And what Manu has been able to develop is certain kinds of tools which anybody can use to make any one of you a scientist. In fact, when Mrs. Murty mentioned about what is science and who is a scientist, I think she explained it such a nice way that I think hopefully it will inspire all of you to become a scientist, not necessarily to believe what comes in your WhatsApp, to believe what you may read in a newspaper or a television, particularly today's 108 channels, which basically call themselves as news channels. So best thing is to validate yourself, what you hear, what you see. To validate, you need to understand the scientific methods. And if the world is going through a major change, there are two types of global changes happening. One is a very high intensity of interaction between people around the world. Itself is pushing the change. Whatever geopolitical changes that we talk about or economic changes we talk about. That change we have seen in India, particularly in the last 30 years since liberalization, what transformation it has gone through, the Bengaluru city itself, the transformation it has gone is one type of change. Another is the major change is the global change is also about the climate change, right? Now climate change is impact, even the economic change impact is different for different regions, for different communities, different people. So each one of us have to develop our own customized solutions. There is no one major solution for this. For this, science is the best way to provide solutions. And that means each one of us have to become scientists. It's not enough that few scientists in Stanford, to help us generate solutions for the whole world. They may provide us an idea, provide us certain tools, not only Stanford, in other places too, and including NCBS. And but innovation has to happen everywhere. To have all of you to become innovators, you need to have an understanding of how science is pursued and what kind of tools you need to validate your ideas, innovation ideas. And for that, certain kinds of tools are required. But tools cannot be too expensive, right? So unless it is accessible, affordable, you cannot be scientist, go beyond human sensory perception. Right? So that's exactly what Manu Prakash has been doing for a couple of decades now. He moved from IIT Kanpur to Harvard, from Harvard to currently at Stanford. Since his undergraduate days, he has been working on developing tools for anybody can do science kind of a approach, vision, and the passion. And the kind of places he has traveled to see what the world offers us to see is amazing. Deep under the ice sheet, obviously he himself has not traveled deep under the ice sheet. He has a core through which he can actually collect the samples and see what is under the ice sheet, or deep ocean, or up in the air. You collect samples from everywhere and see what kind of organisms survive, how are they adapting, right? This whole process of evolution can actually be studied by looking at the natural ecosystem, particularly those which are not observed so far. The reason why they are not observed, perhaps they are not observable with the help of our naked eyes, because we don't have the power to see small organisms, particularly microbes, of the kind of which he is talking about today. Over to you, Manu Prakash. Thank you very much for coming here.
Can you all hear me? Yes? Ulta. I also haven't been in this hall before. I have to say, the chairs are so comfortable. And so if any of you feel like kind of just lying down, enjoying, as Shashi said, please feel free to. No judgment here. You're not in a classroom. You're not in, this is not even a lecture in some sense. It's a rant of just, I want to share how I've been thinking about these ideas. Hopefully they inspire you to do things and take many of these ideas in many different directions, but I would not be offended at all if you take it as a kind of a relaxation. I want to talk about curiosity. And you know, I often look for words and analogies, and the word that I've been thinking quite a lot about recently is chingari. And the reason chingari is just, you know, curiosity is something that lights up your brain, just completely lights it up on fire. And you might think that you are in control, but you actually realize it's so obsessive when you start looking and observing our world in science. Everybody has it. Every one of you is curious scientists, and kind of the claim that I want to make in the opening is really, we are all born scientists, we are scientists. Other people have told us in this ways and forms, you can't do this, you can't do that. We have to preserve that. We have to preserve that for our children. We have to preserve that for our country. We have to preserve this for the world to actually survive. Because at the end of the day, the kind of critical approach that you would take with everything that happens around you is driven by this passion to be able to ask questions. A lot of my colleagues don't agree with this. Sometimes, you know, I have fierce debates about, who should be allowed to be able to engage. And I think one of the approaches that I've taken, which I'll talk quite a lot about, is really sharing sets of tools and getting out of the way. So my job today is to share a few sets of examples and stories from individuals that are actually in this room. Many of them I had not met. They have traveled across the country today to come here and share their own Foldscope stories, and I've never met them. And I just met a gentleman sitting right there from Mysore. He took a train and came, and we met for the first time. And he was part of the program we started in 2011. So I think to me, one of the most important and somebody that I want to thank is sets of people that have engaged in many of these programs, and much of this story is about them. You know, one thing that I want to mention is what really happens in our childhood stays with us. That's my mother. I grew up in Rampur, Eastern UP, very small sets of towns and schools. I had an incredible time at school. You know, you would just explore nature and think about it. That picture I dug up is kind of important to me because I only recently made a connection. That's my brother sitting right there with some friends. Notice he's not wearing his eyeglasses. And my brother had eye problems early on, and I only just realized a couple of months ago that photograph was taken a couple of weeks after when I had stolen his eyeglasses to make my very first microscope out of a cardboard carton from a badminton shuttle. And he was mad at me. Of course, the microscope also didn't work. But it was this realization of the fact that should we just wait for things to come to us, or do we actually have the capacity to create? And I think that capacity to create is in all of us. And many a times he now reminds me that I owe him some eyeglasses. It hasn't happened back. But then something changed, as we all grow up. And the next encounter I had with a microscope after this was in my 10th standard exam, which was this quiz. But ironically, other than the microscope that I had tried to make, I had never seen a microscope. And I was told, and just like all of my friends, is you're going to draw a microscope and draw it exactly as it's in the textbook. And it kind of baffles me till today, because the teacher said, make sure for your examiner to draw it in the perspective, the angle it's there, not the other way around. And so you're thinking about this scenario of something that we haven't even touched and felt. We're supposed to be quizzed upon, we're supposed to understand it. And that sort of kind of created this sense, and when you live with that sense, you realize, life can be tough sometimes in schools, and I'm not going to go into what happened after that. But I think the generalization of all of this is I've lived with this as a fact, that if you think about, there are 2 billion kids on our planet. And roughly, this is the most recent UNICEF number, is around 300 million of these kids live in what would be extreme poverty. Children are the largest group, if you think about, for people. They're more than 50%, although they don't make 50% of the human population that would be in extreme poverty. And when you ask yourself a set of questions around, is science as a universal right? Have we taken the right set of steps? This is, a picture I took in Ghana. Many of you saw what happened to education in COVID. And one of the big things that we've been thinking about as a pillar is to not go through and what do we change dramatically. And here I would say that sometimes I think we are not ambitious enough, because we really have to look at everybody as a baseline. It's not sufficient for science to thrive just in Bangalore. It has to thrive in every corner of a country for it to have the kind of benefits. And to a certain extent, that responsibility falls on many of our partners, political partners, but also on us as scientists. Because to a certain extent, we hold some of these keys, and I'll tell you a few examples of this. Another framework to kind of, you'll see I'm opening this talk with a little bit of depressive tone, and you see, I promise you I will become more cheerful, but sometimes I don't sleep at night, so I need to share my burden. Here's a remarkable painting that I absolutely love. This is by an artist called Isabel Kirkland. The painting is titled Gone, because these species that you see in this painting, every single plant, every single bird has actually gone extinct in the last 50 or so years. We are living the largest, if you think about biodiversity and its capacity, this is being called the 6th extinction. And places like India, we are seeing a rate much faster for loss of species around the world, to a fact that most of these species haven't even been identified, and we are losing them much faster. So this is a massive challenge for the world, especially if we're going to be thinking about solutions that lie in the living world. I spend a lot of time in the ocean. I'm not going to talk too much about it. I wish I could have taken you to the Arctic. I gave a talk at NCBS yesterday, and I really enjoyed it. It's recorded, so you can watch some of that. But one of the big things that we've been studying is how does life impact carbon capture? Everybody's been talking about climate and carbon capture technologies. We don't realize we already have the largest carbon sequestration technology on our planet. It's called the ocean. 30 to 40% of every molecule of carbon that you are emitting with all our human activity, the ocean is thanklessly just taking it in. And if it was to stop, at this moment, the CO2 numbers would go between 525 to 600 ppm. That would be a 3 degrees Celsius change in your temperature instantaneously. And all of this is actually driven by life. It's driven by how life is distributed, small-scale life that we don't even visibly see. In every single droplet of the ocean, there are hundreds of thousands of different species all across the planet, and the way they behave determine how carbon capture works. So, I'm just trying to convince you, if you are, how many of you are going to or about to make a decision to go to college? I want to convince, okay, at least we have one prospect. Think about the living world as much as you might think about computers. And I think one of the important aspects of this is, although the sets of tools have really enabled us to do sets of things, life science holds an incredible key for the future, which I'll talk about. Similarly, I could go on challenges we have in environmental pollution. This is the list of diseases that I've been thinking about. For some of them we actually work on. Ironically, if you just look at this, the number of them that even have any candidate vaccines, that's a very, very small number. We are living in a world, as it changes, the kind of infectious diseases that have a capacity, and COVID was just a taste. We don't have the tools to handle this to begin with. And this is literally with all of the mRNA technologies and everything that's under the sun, and even that's not going to be accessible. We've been working on schistosomiasis for many years, so this is a photograph from Madagascar. We wear up these dungarees and we go out, and most of the locals find that really funny because the kids are playing in the water and enjoying, and we're wearing these things because we are discovering that there are incredible infectious pathogens in these waters. I don't know how many of you have heard of a super mosquito now. So malaria rates have been massively rising around the world because of a super mosquito that's now spreading across Africa for the first time. For the first time in history of malaria, we might actually have urban malaria, like a city like Lagos with incredible density what is known in India, to have falciparum, which is one of the most dangerous strains. Often enough, malaria has been in rural areas, and you kind of end up finding that it's another climate change based induced. So I don't know, are you guys scared yet, or should I keep going on? Because I could skip some of the scary facts, but I just want to make sure that all of you lose some sleep at night today. So I can stop when you tell me, or anyway, it's certain sets of challenges are truly tricky. And I think one way that I would describe some of this is another one of our field sites, and often enough I'm thinking about how do you deliver healthcare when you have very little? And I'll tell you a story about this very soon. That happens to be a malaria clinic that we work in, temporary clinics that are set up for people to come in. And I've spent a lot of time in Africa, but I can tell you very many things that I've seen in India, especially in the rural context. We have to do a massive degree of improvement for primary health care to actually improve across India. So one thing to think about this is, why the living world? A living world is of course beautiful, and we'll spend a lot of time talking about curiosity and beauty. But the living world is also a technology of abundance. Because of the capacity that you can take what you have, you can share it, you can grow it, you can literally grow it in your garage. And it's been ironic that the most expensive sets of solutions that we find that are built with the living world are not frugal, while the living world by itself is frugal, because you get the blueprint, you can literally replicate it. There's very few airplanes that self-replicate. But frankly, I could give you a strain of yeast that's going to self-replicate that can cure a particular disease or be producing insulin. So why the hell have we created a world in which these sets of technologies have been locked for a long time? And frankly, we have to answer this question. It is a societal context. It's about how we distribute technologies. How do we take a stand on open science? It has nothing to do with technology at the core itself. Out of the 17 SDG goals, 11 of them have solutions that are primarily, can only be found in the living world itself. Okay, so I think I don't have to tell you that we're not on track. I'm not going to spend time, this is a political side. I mean, the world is in a mess with what's happening in regressive nature of what we are doing to science currently in the United States. So, all right, so that was the depressing part of the slide. Tech, you can shake it off a little bit, or you can mull over it for some time. I find it very hard to shake it off because every one of these stories that I told you have humans behind it that sat me down and told me these stories, and that leads to when I'm working in the deep ocean and I pull out a fish or a squid from 1,000 meters and it's completely filled with plastic. In the deep ocean, we're finding plastic. So it's, every one of them is hard to shrug, and one of the big things that we've been thinking about for a while is how do we take an approach that allows to focus on tools and sharing those tools so many, many more people can engage in solutions. Because frankly, ideas are everywhere. They're in this room. Of course, you have to have an empathy for these problems, but also you have to have the right set of tools. So we've been calling this frugal science, and I teach this class now for 500 students around the world, all across the country. It's completely free. Any of you can join. We've been doing this for 5 years, trying to build this philosophy based on a decade of work for specific sets of tools. And then a student last year joined from Sudan. Actually, 2 years ago. His name was Marwan. And we were starting education programs with him in Sudan. He had been thinking about education. And in the very last day of class, he said, Manu, I now understand what you're trying to do. And in Arabic, we already have a saying for this in Sudan, which is we say, don't create a pen that doesn't eradicate ignorance and shame. Pen is a symbol of technology. And what he meant by that is we are the creators of these sets of technologies, and unless we can be very deliberate in how we would implement these tools, how we as scientists would care about these tools being delivered to people, you would inadvertently create sets of technologies that don't solve the sets of problems that you care about. And ironically, the day I had the chat with him, we hung up, and then I read the news 2 days later, war broke out in Sudan. He had to leave the family. We had to completely scratch all the plans that we had. And we've been in touch for a while. He's okay, but it completely transformed. And so, of course, there are all of these other sets of factors that are at play. So even in peace times, we cannot really implement the sets of solutions. Malaria hides in many of these conflict zones. You can think about many of these problems have geopolitical context itself as well. Okay, so I think I'm going to give you a few sets of examples of how to try to think about this. I don't claim to have all the answers. We have a lot more number of questions. These are global problems. We don't have enough scientists on this planet, enough doctors on these planets to deliver the kind of solutions, period. And so community health workers are what comes to mind when I'm thinking about kind of a citizen scientist as an approach to this. It has to be an inclusive approach where we have to take this that everybody is allowed to engage in problem solving. They bring something new to the table. And now the question I was asking is, has this been done before? It's been done many times, and I'm going to give you one of my favorite examples. So this is time for a quiz. I want to check who all is sleeping. How many of you can recognize what that object is? Raise your hand if you know what that object is. So that's fantastic. I see some young people. Can you shout out? What do you think that is? Yes. Sputnik was like a basketball-like object that transformed our world. Flying in space, everybody wanted to see Sputnik. And of course it caused fear because everybody was worried that, oh, now we have surveillance ongoing, although Sputnik had no cameras or anything. But it had a radio, and it would transmit. If you go to the right time, you could go and record the beep. And I'm told when I read many of these sets of accounts that people would go down, bring their family out, and just watch. And it was the very first time you could feel something really that's out there that felt extraterrestrial that had been created by humans. But the other part of the story was many of the national defense organizations panicked. Because when Sputnik was launched, we had no capacity to actually observe our skies. And there was an astronomer, Fred Whipple, who had perceived this problem and realized the sky is really big, and he started what became Operation Moonwatch. Literally $10, $15 telescopes shipped to thousands and tens of thousands of individuals like this around the world, competing with some of the state-of-the-art $100 million technologies, and guess who won? For the next 60 years, these individuals individuals like you actually track not just Sputnik, but every single satellite. The fact that we have the only supernova that's been ever seen has been by an amateur astronomer who was fixing a camera on a rooftop in Australia is primarily because of these programs. This became really an absolute craze, and for the next 60 years, this was the primary data that we have, very quantitative data collected by people around the world. And then when I think about Fred and his sets of programs, what comes to mind is community health workers. You could argue that the world's largest citizen science program are these community health workers. They have a high school degree. Many of the ones that I work in, this is in Liberia, are 4th grade, 5th grade. They have incredible set of assets that they bring to the table. They'll work with people. I mean, this girl, much younger than me, is with a backpack heading out to a village in Liberia where she knows there is Ebola. And the kind of protection she has is just that little backpack. And so one of the things that we think about is what are the kind of tools that we can provide these individuals. So that led to this philosophy of we can work with individuals, but we have to provide them the right set of scientific tools. And one of the tools that I'll talk about is Foldscope, but I think one of the things that I also just want to show you is although we build and design tools for specific purposes, the true purpose is to just bring joy to people. And that's kind of reflected. What I love about that photograph is anybody who's seen through a Foldscope, and some of you have done that, there is a self-detector that built in this technology. If the person is smiling, you know they got it. If they're not smiling, that they need to keep looking and the microscope is not working. And that is really one of the primary goals when we think about these sets of tools, because at the end of the day, you might not have an absolute solution to that problem, but you've actually created this capacity for them to start digging and capacity for them to start engaging. So we're going to spend some time. I'm going to take you to a few countries around the world. Also, I forgot to mention, my talks become exponentially faster as I go because I then look at the clock and I realize, oh my goodness, I want to cover a lot. So I apologize for it, stop me at some point, and then some of the technical details I can also cover right after. So we've spent a lot of time and realized that science belongs in the field. The way you identify these problems is you spend time with people. And I remember an individual I met in Uganda. I was a first year assistant professor. You know, it's a powerful moment. You get your own lab. You know, I can buy whatever I want. It's like a kid in a candy shop. And I went in this very proud way. I was spending a lot of time in Uganda and Thailand at that time. And then this individual who had trained 30 or 40 years of all of these microscopists and medical doctors said, Manu, come back to me when you can do diagnostics under a tree. And that stuck with me, and I'll talk about kind of how do you deliver healthcare under a tree and many sets of tools and technologies around it, because he had this sense. And I just got back from Nigeria. We ran a massive program for kids in Nigeria for schistosomiasis diagnostics. When we arrived there, either we are operating in a classroom that has no electricity or we are working under a tree. So now, after 15 years of doing the work, I actually understand what he meant by doing that. And then that's also been a technological challenge for us. How do you deliver first-rate healthcare, first-rate sets of solutions that can be operated without any electricity, without any infrastructure? On the expeditions, you know, I'm not going to spend some time, but this has been really a passion for ours to take many of these tools, not just us, but others, out to sea to really be able to do the kind of global environmental surveillance. So I'll give you a few examples of that. I snuck in one video for you to just get a sense for what it feels like to be operating in the Arctic. We were trying to measure how much carbon sequestration is happening under ice. It's kind of scary because this ice is quite thin, and then polar bears show up sometimes, and the strict instructions are if polar bear is in sight, you rush back, and your experiment is delayed 7 hours, and then after that you come back, because polar bears now, there is no food, so they hunt researchers, which, you know, I don't know, is that a good story to end life at? I don't know. I debate sometimes, although going down that way would actually be fun. Okay, so here is a few sets of examples. You know, we've released dozens of tools. Many of them have scaled. I'm just going to now give you a brief summary of a few of them, but I'm also just giving you this as a menu that you can build upon any of these tools. We open source almost everything that we do. So many of you who are inclined to engage in technology, in production, in scale, what I'm really excited to be here is the fact that many of you have ideas in how to take these tools in directions that I can never imagine. And that would be the real outcome of a public engagement like this, is when many of you see utility of these tools in your own areas, or you're inspired to build your own solutions on top of this. So all of these platforms are completely open. We do much of this science in the open primarily for the fact that we realize as a group we're small, but we're mighty because we build communities on these programs. And I'll give you one or two examples of those communities. Okay, so let me tell you a story of malaria that starts in Kalahandi. Sister Akinas invited me to spend some time in Kalahandi, and I was talking to a few people. How many of you know where Kalahandi is? Okay. How many of you been to Kalahandi or area? Okay, only Sudha and a few people. It's a remarkable area. It's a tribal area. It is one of the big pockets of falciparum, one of the most dangerous mosquito malaria strains. Now the challenge is India has this mandate that we're going to eliminate malaria. We're not on track. To be very honest, we're not on track. Vivax is just really difficult to be even tracked to begin with. And even in these sets of pockets, one of the things that I have struggled with is look at the numbers, by 2017, there was a massive decline in number of malaria cases. And then 2024, look at that, 263 million. All the technology, every possible global organization pumping money, that is the malaria numbers that we're dealing with. And the curve is going up and nobody can tell you why it's going up. Talking about fundamental technologies, when you mention COVID being an ecological problem, this is an ecological problem that we don't understand. Simply because for every one of you, there are billions and billions of mosquitoes out there. And I'll show you, we'll kind of spend some time with mosquitoes. How many of you like mosquitoes? A few of you like mosquitoes. We'll spend some time with a mosquito. So I go to Kalahandi, I was excited, we spent some time, we started training people, and I spent a lot of time with Durga and Prem. And I want you to become Durga and Prem for the next 5 minutes. That's Kalahandi, one room hospital, that's it. In the entire area, that's the primary center, I'm already in the diagnostic center. In that window, somebody would tap, and Durga and Prem for basically 9 to 10 hours a day will sit on a microscope looking through slide through slide through slide. India has the largest microscopist in healthcare industry in the world. It's remarkable. 95% of all specimens in diagnostics that are seen in microscopy are in India. So India did not dissolve our capacity to do microscopy, which is amazing because now Africa, that was not the case when many of these RDTs came along, and now we're struggling because we have a lot of resistance that has occurred. So I want you to really take a moment and see what your life feels like if this is what you do day in and out. It's a moment of pride. They get trained, they're masters at this job, but when you do this 24/7 all the time, it is very difficult to deliver the kind of care. And they would much rather be working with patients per se, so they would spend between half an hour to 45 minutes on a negative sample. So you can understand the set of challenges. There's absolutely no capacity for molecular samples. It costs 5 cents to do microscopy. RDTs many a times cost much higher. The specificity is not high. And that's primarily because the primary strain and the primary assay that we have been using in malaria was actually invented by Juan Gimsa in 1904. So talk about evolution of technology. We're still using something that is more than 100 years old. And we know so much about basic science. We can improve this astronomically, but we haven't had the tenacity to say that let's actually change this. Let's try new things. And I'll give you an example of what we've been trying in that space. I love this photograph. I'm assuming you all know who that individual is. There is a little debate, by the way, for whether what is being inspected is actually a malaria sample of Mahatma Gandhi's own blood, or is it actually a sample from Leishmaniasis, because he's very interested in many of the infectious diseases. And I've been trying to find the archives. Yeah? Leprosy, yes. And so I want to know whether it was malaria or leprosy, and it's kind of it's been a challenge. But the other thing that I also find fascinating in this photograph is you can see he's using a microscope that was, built, and one of the things that's valuable in these sets of images and photographs, it kind of reminds you that very early on we have had kind of the adoption of technology as a way to move the world. Ironically, India has one of the largest microscopists. You can see the sets of numbers, 200 million slides. We have absolutely nothing in the digital space. And I live in San Francisco, and I can tell you it's a scary time. The streets are covered with these self-driving cars. Almost, it feels like 30 to 40% of these cars are self-driving, and on the other hand, we have these tremendous challenges that we could apply all of those technologies to, and we could solve them right away. But then again, we choose the wrong problems to work on sometimes. And again, in my opinion, that is, when I look at the kind of a global context for this, we can realize that a remarkable number of tools that have been built for other purposes could actually be repurposed. So here's an example of what we ended up doing. We started asking ourselves a question: what is a minimal system we could build that would completely automate it? It is only possible because gamers, I don't know if there are gamers in the audience, many people like playing video games. That led to faster GPUs, that led to the machines where we can do this, and I'll show you the evolution of these tools. And now for the very first time we have an instrument that we've been deploying across several places in India, but actually 25 institutions across Africa to do something very simple. So this is the actual tool, and I'm just going to give you a quick video for this. It's the first time we can do completely autonomous diagnostics of malaria out in the field on a cell phone charger. Between 5 minutes or so, this tool has a capacity to image 20 to 30 million cells. We can pick up very, very low, 5 to 10 parasites per microliter. And one of the things that starts to happen is, this is a quick video of how the tool operates. And I think since I don't have time, I'm going to skip this. But anyway, trust me, you put a slide, you walk away, it reads, lots of fun machine learning. And then something interesting happened. Again, going back to, we built the tool, it took us 7 years to build this, we started implementing. And a group from Nepal reached out. They said, we have sickle cell anemia here, could your tool do this? And so that's the tool, it leads to detection and diagnostics completely autonomously. Actually, no, let me tell you a story from Assam before. The program was working everywhere, but we had a problem with some of the science collaborators who were working in Assam, that every time they would send us a slide, it would not look clean. And I was like, can you tell me how are you making these slides? And then they sent me these pictures. So this is literally field programs, because malaria is a field program, and literally sitting on the street, somebody's making smears. And it dawned on us that something that we have been doing for the last 200 years, making smears, is actually a problem. So I don't know how many of you have made smears. This is what you do when you go to your first undergraduate medical training. You take a drop of blood and you just squeeze it. You have to get it right. And then you train and train and train. I don't know how many of you have done that. I can't do it still. But we asked a question, mathematically, what are we actually doing? Turns out this is what we're doing. You write a single equation and that can tell you what's happening. But for the last 200 years, nobody had written this equation. Now, when you can do the science for it, for a 3D printed object, it has a little valve right there, 5 cents of a valve, 17 cents of a syringe. We can actually build an instrument, has no electricity, that can completely automate smear making out in the field. And actually all the gizmos that we built for malaria diagnostics would never have worked if we had not actually built this tool. So I know it's going to work because this is a video, but you can try this too. And all it is doing is essentially using air as a spring, a valve that is in your bicycle as a leak, and you make perfect smears, and then you have 30 million cells on this object. And we've been implementing this as a program, and now healthcare workers around the world, currently we have this in 50 countries around the world testing, and they're starting to now build databases and smears that are associated with a simple tool. They get digitized. So now back to Nepal. Now Pranav wrote to me, and he was really interested in sickle cell anemia. I had never worked on it. We said, we'll give you the tool, you run the clinical trial in Nepal, you do the clinical work, and within 7 months, then they release the paper that shows that, okay, you can build a machine learning model to do sickle cell anemia. And then we asked, why stop at sickle cell? Could we do TB? I mean, TB is a massive challenge. We partnered with another collaborator because I think one of the challenges with TB is also detecting live TB, because it's an infectious disease, and it turns out we can do this for TB. We can actually do this for sexually transmitted diseases, and at this point, there's roughly around 30 or so diseases that we are targeting. So it's a little bit like an app store. Anybody can build these models. There is a standardized imaging instrument. Anybody can have these sets of instruments, and if you have an interest and you have clinical expertise, you own the data, you build that application on top of it. And again, it goes back to thinking about how do you share technology that other people can also have an ownership in it, and they're bringing the kind of expertise that's actually needed for it. Now, something happened also along the way. I'm going to show an object that I know many of you recognize. This is a very important object from the history of Bangalore. This is Raman's instrument. If you haven't seen it, you should take a look at it. Raman was thinking about frugal science way before anybody, and I'm told for only a couple $100 rupees, he actually built his first instrument, and then of course he's improved it over time. But it's a very powerful technology. We've been using it to actually detect sepsis, many of the other sets of diseases, and we can do now label-free diagnostics of some of these scenarios. So really very precise bacterial species can be identified purely from a Raman spectra. I had to mention Raman in Bangalore, it's very important. Okay, so this has been growing as a program. It's become kind of an app store. And one thing that I'm quite excited about, to find clinical partners in India to take this in any direction that they want. In parallel, a lot of scientific institutes started reaching out saying, oh, we don't even have fluorescence microscopes, so could we use these tools in our basic science? And that led to another program called SQUID. So if any of you do science, now this is the big brother of Foldscope. I'm going to talk about Foldscope very soon, but it's important to understand that this big brother now allows us to do any kind of automated microscopy, all in the complete open source platform. And that's, this is the types of open source platforms that we build right now. And anybody now, going back to programming, can actually program complex assays. And I'll give you one example of why this became important for us, because we were thinking quite a lot about Ebola. One of the realizations that we made, so now this is Monrovia. Many of the patients that got infected with Ebola that survived didn't have any vaccines, but we don't understand why. So you have to go back to that population, understand that population, but many a times communities, there is absolutely no sample collections happen, but you don't really build the capacity. So now this is the first immunophenotyping station that we put together in Monrovia, where now the science is also happening exactly where the problem is. And it's a very important thing to be thinking about for clinical operations, to really deliver that clinical capacity as far as possible, because that's when you inspire many of those students to actually engage in those problems. Okay, so I've talked about diseases, and since I'm running out of time, I'll just mention this. I don't think I'll have time to go through much of this. The other problem has been molecular diagnostics. A lot of work has happened in India. It's a really good place to be right now. ASHA workers go out there and do diagnostics for a while. We've been thinking about molecular diagnostics for a while, and we were inspired by essentially a ballpoint pen. And we call this object D3 or SnapDX, which is some of the physics that's actually used in manufacturing ballpoint pens. We can use that to manufacture essentially a molecular diagnostic test. So now this is not imaging. You can essentially identify individual, very specific sequences and molecules. It uses hot water. And because of that, I have a cup of coffee. You pour, and if you can make tea, you can actually do a molecular diagnostic test. It uses the heat of that water to drive the reaction. There's a couple of clever physics inside it, and you read it out like a ballpoint pen. And again, one of the things has been, it's a platform technology that allows us to implement many sets of diseases. As you'll see, I'm going to go much, much faster on these sets of, what's been important for us is to also not just be thinking about the tool, but the manufacturing of that tool. And so one thing that led to be is asking a question, could we manufacture a molecular diagnostic test in a shipping container? And the idea is that you could locally manufacture and put the manufacturing capacity closer to where the diseases are. I mean, the number of infectious diseases is very large, so you have to build that capacity, and so this is what we're building. The current partner, we built one of these trucks on Stanford campus to show that we can do this, and currently there is a partnership with Institut Pasteur in Dakar where we're building this in Senegal to just prove the point that you can bring manufacturing very quickly for molecular diagnostics. And again, a lot of work of this has happened actually in India itself. Okay, let's briefly talk about mosquitoes. I know some of you mentioned you like them. I am going to play an audio, and let's hope that audio actually works. First of all, you can do a back of the envelope calculation. There's a lot of mosquitoes out there. You could do a calculation for Bangalore. It would be fun to know how many mosquitoes per individual person. That number is very large. That's why these diseases like dengue are very difficult to fight. And one of the things that we've been thinking about is how do you go about tackling this? Many of you know Ronald Ross here in India discovered malaria in a mosquito. This happens to be a postdoc. I don't know, how many postdocs in this audience? Okay, postdocs are the backbone of science. They do the hard work. I see at least one person. Oh, a couple here. That is a postdoc that has been given a bucket of mosquitoes, and he has a tweezer in his hand and a microscope. And his task is to tell what species and do a count for every one of them. And this is the reality of our technology when it comes to what we're actually doing in ecology. This cannot scale. And this is one of the reasons you cannot tell me how many species of mosquitoes are there in Bangalore, because we haven't built the infrastructure. We got excited about this problem, and I'm going to play an audio, and you're supposed to tell me what are you listening to. Anybody who figures it out can just shout. That's good. This is the deadliest orchestra on the planet. This is wing-beat tones of 40 of the deadliest mosquitoes, and I was playing with a musician friend of mine, and we paired them to turn this into a music note. If any of you want, you can use this as your ringtone. It's a fantastic conversation starter anywhere. And why did we do this? The fact that your ear can hear those tones means that you could acoustically detect species of flying mosquitoes. Because they mate in flight, they actually emit a very particular frequency, and that's the buzz in your ear. You can train your ear, but better, you can actually train a cell phone, and what we showed is actually using just cell phones, a $5 flip phone, which we have billions of. You can build the world's largest mosquito tracking network. And now DeepMind has gotten interested. They released another version for this for now. Why stop at mosquitoes? They can actually do many insects. Completely open as a platform, and it enables now. We've built many programs training vector control authorities across the world to train them to actually train students, and I'll show you a video now. This is a picture from Madagascar. We do a lot of work in Madagascar. This is Rana Mafan. I'm in a classroom, and I'll let you just see what happens when you teach kids about science that they care about. So they're buzzing with excitement because they finally feel, and then we showed that these kids can make a mosquito map much better than a few handful of scientists across many of the villages in Madagascar. So again, another example, when you give power to people who actually suffer from the sets of problems, they can do remarkable things. I'm going to spend 2 minutes on period poverty and kind of a challenge that we're facing currently in menstrual health. This is the map of the world, and there is a worrying problem with climate change. Many of you know cotton belts around the world. Cotton and timber are the primary things that provide help for menstrual pads. Cotton is a very water-hungry plant. If you actually look at many of the challenges across in South India where there is no rainfall, many of the cotton farmers have a very difficult time, and it changes in demand. And timber, there is none of, other than many certain parts of North America. And then when we looked at Africa, one of the challenges was it's a very dry part, and we don't have anything to replace cotton. You need an absorbent to make, unless you use polymers, which then cause massive amount of pollution. And we were starting to do these calculations, and it occurred to us, why don't we expand and search for the biodiversity to find a replacement for cotton? We tested 115 different plants from around the world, and surprise, surprise, we actually found a cactus that grows across the world, in which it's called sisal, that we can use to now beat cotton at its game. We can produce a material that is, and we use cotton candy machines to actually make this material. Let's skip that. And the process to do this is a little bit tricky, because cotton is the only plant that gives you pure cellulose, but other plants have these mixed lignin and other sets of things, and we use essentially a process that's used in a gut of a termite to develop this material. And now this material, you can actually look, it's much, much better than cotton in absorption. And we actually just, last week we started our first factory. It's a large scale factory to make sisal and sisal-based menstrual pads. And again, one of the key factors around this is the biological resources locally available across Africa. You have the capacity to manufacture the sets of machines, and you also have the capacity to build quality products that are built from local raw materials. Okay, so I think let me skip oral cancer. I think there's a couple of threads on oral cancer that I would love to talk about, but I'm going to run out of time. I don't want to talk about COVID too much, because that's still too dark a time, and for some other day. Let me switch to environment briefly, and then I promise I'll stop in 10-ish minutes. Okay, so I don't know if any of you can identify, that's Lake Chilika. We had arrived at Lake Chilika because of a request of one of the ecological centers, and they were having a hard time predicting what's happening to local fish. We started building these microscopes. Now this is called a planktoscope. It's now a microscope that's made out of bamboo. It allows us to essentially image every single drop of water to tell what's in there. All the instructions are online. We started building and sharing these. It cost a couple $100. And this is the type of data sets that come out of it. And so if you think about water and access to water, we have very little monitoring on the aquatic kind of a context. But completely automated, it allows us to essentially build these data sets of beautiful species, and the word that I use is ecograms, that you can take an ecological photograph of an aquatic place. All done by community members around the world. These are some of the ecograms from sets of communities. When we shared this online, only by just instructions, people started building them, and there are many of them actually in South India currently. This is a large community that has grown, and many of them are really passionate about water and access to water and thinking about how to actually monitor water and water sets of resources. It's now allowed us to also grow the kind of data sets that actually exist in the oceanic world. Now there are versions that we can tether and float in the oceans. This is the data set coming out now from an in situ planktoscope that you just dangle, and it will beam cell phone data. You can connect it to your TV, so rather than watching TV series, you can now watch what's happening in your local pond or your well. But it really allows us to understand the complexity of these ecosystems, because these organisms don't live alone. Any of you can build these sets of tools. It's completely open source as a platform. And then there's a lot of AI that's been built, and actually now I'm going to switch to the last topic that I want to mention, which is about Foldscope. Let me get a show of hands. How many of you have used a Foldscope before? Wow, okay, so at least 10% of you. That is super exciting. This is sort of an object that we released several years ago that has grown quite a lot from a sense of a community. I'll tell you a very brief story, but for some of you who haven't seen, a whole bunch of pieces of paper, an objective allows you to make a microscope that starts to compete with many of the traditional microscope. At the heart of it, when we built this, I was very proud. We wrote a paper, and again, as academics, you have to learn lessons the hard way. I woke up the next day, the paper was online, and nothing in the world changed. As an academic, it's a really damning thing because you work very hard and you say, I put my work there, it's there now, and the world is going to change, and nothing changes in the world. And I think this is a lesson that I learned. And there was a funny way I learned this lesson. So first of all, the microscope competes with all kinds of microscopes that you can really use. We started sharing this microscope with people, with sets of communities. You understand what this student is trying to say. And at that time, we built this microscope primarily also for specific problems, but to just build curiosity and joy in people. And again, I mean children at heart, and so that includes all of you. It connects with cell phones, it allows you to collect kind of data, but the most important aspect of this was really what kind of questions people would ask. And suddenly a massive alien world started opening to people that had never had the chance to see this microscopic world. It can take you to places which make you uncomfortable. I just looked at my cat's mouth and oh my God, I'm never gonna let it lick my face again. If you love your cat, don't look at what's growing inside its mouth. And literally all of this data is coming from users from around the world. We started scaling and sharing this set of a tool, and I'll tell you in a second how we decided to. But truly there is an alien world right under your shoes, right under your nose, right on you. And the only way to experience is to not read about it, not to watch videos, but go out there, pick up a microscope and observe. You can use these instruments, you can use any instruments that you want, but frankly, the most alien thing that I can tell you and share with you is in the small scale. Every single time I look at something, I am surprised. I've been doing this for the last 15 years. Every single moment I look at this, and this is sort of obsessive, you start obsessing over everything that you look. That's actually immune cells inside a beetle's leg that is alive. And that's glass krills banging their head. There are organisms that are completely transparent. When you think about, imagine all of us being transparent. How cool would that be? One of the things that starts to happen, this happens to be my wound. I got poison oak and it's wound on my skin healing. This happens to be embryos developing, and I mean it's insane in terms of what you can start seeing under a tiny little microscope. All of this data comes from an object that costs $2. This actually happens to be an unknown species that we are yet to identify that has a symbiont. This is where spiders come from. And again, all of this data collected by individuals that we have never met. We shared the sets of tools. This is what is happening in your kitchen sink. So again, also don't look at your kitchen sink if you are, this happens to be ticks. And again, this world is beautiful and surprising, and the sets of tools have been used around the world. But I want to tell you why, what kind of irked us and got started. I told you this story, we published this paper, I was super excited, and then a friend sent me a text message, hey, did you notice some fairly well-known gentleman, that I'm not going to name here, but you can read the name, had a tweet, and he said, a $1 microscope, ? Interesting, ... And I was like, what the hell, why would he put a question mark? Is he questioning what we have done? And then, I came back to the lab and I was talking to my student, Jim, who's done this work with me. And I was like, Jim, we have to do something about it. We have to respond. And the way we responded, we said, okay, if the world doesn't believe us, we are going to give a free microscope to anybody who asks. And it's very rare. And today you get a free microscope. And actually now we're building a massive distribution network. I wrote this on my lab website, and a lot of my colleagues were puzzled. It's like, what the hell are you doing? Like, you're supposed to do science here. Is this science? I was like, yeah, I have to prove to the world that actually this is possible, and I don't know what people will do with it, but it is an experiment. I want to find out. It's a social experiment. I'm not a social scientist, but I'm going to do this. We got 75,000 emails from around the world, 150 countries. We spent $300,000 of our money from my startup to actually ship to Congo, to Brazil, everywhere that we could find, massive, and a few of the people from that Foldscope first program are actually here. We would have never met if, and so we shipped around 100,000 instruments. That led to lots of people. There used to be politicians that knew about science. And one of the challenge that it sort of brought to us is it's grown now over the years. It's been 10 years we've been doing this. This is the largest community. 3 million kids around the world use this tool. 500,000 of them are in India, and we were able to implement in India because of government partnerships. We did a partnership with DBT, with Vijay, and I think that was a pilot, and now I'm really excited about scaling this from 3 to 100 million kids. Remember, I told you the number, 2 billion kids. That's the one that we have to actually address. This photograph is, remarkable to me. This is from a village close to Vellore by Mo Pandirajan, who unfortunately is stuck probably. Mo, are you here? He could not come, yeah, so he's stuck in a train somewhere. He sent me this photograph. This photograph makes me so happy. You can see the joy. Who tells you that science is not exciting? I can hear these kids in a photograph. But it also makes me very sad. Look at the line, and there is one Foldscope they have, and they're sharing, and that's what we have to change. So although we have reached 3 million kids around the world, we still have 2 billion to go. And I often feel that in fields like education, in many of the social contexts, we're not ambitious enough. We have to set the goalpost as high that every single kid, if there are 300 million kids in this country, we should actually be addressing that need at that level. And lots to talk about and how to do that. Many of you have to be involved in being able to take these tools. This cannot be done just from far away and externally. Mentors are required. This is another photograph I love. It's one of my favorite photographs. We manufacture microscopes, we don't manufacture mentors. What I love about this picture, these are parents that are not sharing the microscope with their own child. You have to be that parent, because science transmits through osmosis. When the child realizes that you are obsessed by something, they find oh, there must be something interesting here, I want to get in. So unless we can be the children and we can be the model for how we want to perceive curiosity and share curiosity, we'll never be able to solve this. Okay, I'm going to go much faster now because this program is quite large. I could tell you literally thousands of stories about this. Let me just skip all of this. There's lots of tools that have been built. This is a large community. That happens to be Liberia. We run programs. This is Dayalbag, 5,000 kids arriving in a tribal area. And much of this has only been possible because of very passionate people like Anupma and many of the other Foldscope fellows that we only have met once or twice. We've been able to translate these tools. They travel across the country, and I'm going to share a story of one of them. That happens to be, that's the age group. All of this data is online. It's archived. We now have a genomics program that we're building so we can build the biodiversity capacity. This is another tribal area. Farmers being able to use these tools to actually detect pathogens. Ideas that I would have never thought. We just had to share the tools. I have no background in agriculture. I have no clue. And then suddenly somebody writes to me saying, oh, we have identified 13 different pathogens for these sets of... That's the power when you share a platform and let other people build on it. And I think much of the ownership of these ideas belong to the people in the community itself. This happens to be the Albag. This is an old, roughly around 1,500 publications. Out of them, 1,000 have actually come out from India. There are new species that have been discovered. There's a new game we're releasing that I'll tell you about. How many of you love Pokemon? Or at least know about Pokemon? So now this is Pokemon, but for real, because the microscope actually has playing cards, but the card has a little hole. And so if you go collect something, which I could just do right here, I could go in, and I was noticing that there's actually some viral infection on these plant leaves. So I'm going to do I look in here, I find in, I touch this, and suddenly now I have made a sample that was collected in the, sorry I didn't ask any, I just broke a little plant, but I get excited. Now this object acts as an archival object of what was growing, what was happening here. And now when millions of kids walking around the world have their real Pokemon cards right there, We have this massive sharing network. This gives you a community-driven way to map biodiversity, a kind of biodiversity that has never been seen. Because if you want, I'm sure many of you are aspirational scientists, here's an idea. If you want to name a species, go for the small stuff. All the big stuff has either we've killed it or you have actually already been named. And I'll tell you kind of a challenge in that space. So this has been growing. We've built a partnership with Nanopore and a couple other sequencing stations to build sequencing archives. This is another sort of a community workshops around thinking about agricultural pathogens to be detected. That was in Andhra. This happens to be the first animal to do microscopy. And it's, it's partly a joke, but this is Gurek. He's the first sailor to cross the kind of Canadian Passage. And we engage with adventurers. I don't know if one of the adventurers that went out to Himalaya and found 15 tardigrade species is here. There's a student from India who took a motorcycle, took a Foldscope, traveled across Himalayas. I wish I could do that. And then identified new species of tardigrades out in the cold, all using his Foldscope. And one of the things, this is another individual, Dr. Kirti, she identified a completely new species of cyanobacteria in Pune from the municipality water. And of course, that's where you want to look. It's a great place to find new species, but a species that has never been identified. And I want to mention a shout out to an individual that I really aspire to. This is Mo Pandirajan. Lives close to a village in Vellore. He had written me an email in Tamil. I don't speak Tamil. This was in the first phase of the program. I translated it. He said, I run a 10-15 children's school. I would like some Foldscopes. Would you send? I send it. And then I started seeing all of these kind of on our community website, his post and the work he does. We started engaging, we became friends, I started shipping some microscopes to him. This is just one individual working alone in a rural location close to Vellore. No connectivity with anybody else, just one individual. And then 4 years later, I get an Excel sheet with 110,000 names in Tamil and another sheet with 12,000 names. And then we were like, what is going on? He said, oh, by the way, I just crossed 100,000 students that I have trained. One single person has trained 1/5 of all of the Foldscopers in this country. Think about it. Now, and again, I really wish you could have met him because he's a humble person, but while you will be talking to him, he would actually teach you how to Foldscope. And he goes to weddings and he's teaching Foldscope. He goes to a railway station, he's teaching them. And one of the way I finally figured out that he did it is became Facebook friends with him. Every day he's running a workshop for 50 to 100 kids anywhere in the country. And and again, many of these Foldscope fellows exist. I've been thinking quite a lot about how do we support this citizens approach. What if we had 1,000 Mo Pandirajans? What would it do? I mean, it would transform our country's capacity to actually explore this world. And this is very important. All of us cannot do this. You really want people to be passionately taking this on as something that they care about. But there is nothing in his CV that would tell you that he's the right person. So the way we do this is we thinly lay out the resources and they rise from the bottom. That's the only approach that I know, is that you share the resource with everyone, and anybody who's passionate, I mean, many people say they're passionate, but when you quit everything and you care about children so much, you actually end up taking this. And I think we need these kind of individuals. We need to urgently support them because they are not in our academic systems. Mo has no B.Ed degree. He doesn't have, and now he's gained a ton of knowledge, but this is not where he started. His root is that he deeply cares about engaging with children, and he's as curious as anybody I have met, even on any academic circles. So I know we are out of time. There's lots of crisis programs that we built. There was another individual, Sonali, that was going to come from Mumbai. She built another program called Gully Well for mental health now in Dharavi, but using microscopes to study mental health and let the kids who have no access to nature. This is a program in Iraq. Many of these kids are living in environments that all of us should be shredding. We should be worried about what are we actually doing. And one of the ways to actually, in urban environments specifically, is that the microbial world give you this kind of an escape. We have built many sets of programs in juvenile prisons, many sets of conditions where you really completely kill creativity. And it's very important to be thinking about these sets of programs. I have so many stories to tell. I'm going to run out of time. I will just skip to something that I want to say. I think I just want to mention two things. One is we could have a Chandrayaan-like moment for our country if we made the largest grassroots network and we showed the world that we can build biodiversity maps and we care about biodiversity. It might be that we are the largest biodiversity hotspot of the world. It's very important to be thinking about the past expeditions. If any of you know the history of the Hardwick collection, were all driven by colonial powers. Many of the painters that have described and drawn these species that are in National Museum in London are actually Indian painters that have never been named. The titles say an unnamed painter from India. That's it. Although Hardwick has like 30, 40 species just named after me. Hardwick was a colonel in the army, just going around and collecting. So we have really, that's how much of the life science and the natural history in this country was discovered, sort of. We have the chance to change the narrative. We could change the narrative if we could really build a people's movement in being able to caring about biodiversity, engaging the communities that have built the kind of knowledge, and guess who's going to protect it? Those would be the communities that would actually be protecting this. So I'm very excited about this in the sense of rethinking what it means to do exploration in the current context and actually realizing and facing and showing the world what the history has actually done. Let me end with sort of a sad note. I want to share this because it was Teachers' Day and I've never said this publicly or so publicly. I've told this to my friends. But I know I'm out of time, but let me share this story. I went to IIT Kanpur. I'm deeply grateful to what IIT Kanpur gave me. I was pretty bottom grade. I barely passed. But I loved grad school, I loved science, and people were able to give me these set of opportunities because I was spending way too much time doing other sets of things which were relevant and important. I had proposed a thesis which was about wanting to write a computer program that would draw like a child. And most of the professors are like, what the hell are you talking about? This is a computer science program. We don't do psychology here. But there was an individual that heard me. His name is Amitabh Mukherjee. And he said, there's something interesting here. Let's think about this. I think we can build this in the context of AI, and it's actually really interesting. We don't understand how a child draws. Such a simple thing. And he convinced many other people, and I passed, and so here I am. It's good. I'm thankful to the institution to support that. But without that individual, I don't think I would have had this realization that unbounded curiosity will actually be supported. And so in every one of you, you have the capacity to support unbounded, just uninhibited curiosity, and many of you do this every day. The reason I'm sharing the story is it was Teachers' Day yesterday, and a couple of months ago, I haven't been to IIT Kanpur since I graduated for whatever reasons. I fell, I'm not very good on email, as Neha knows, so I don't connect with people many a times. And my kids found a letter in my house which Amitabh had written me, but I had never read it. The letter described sets of things, and my daughter, who's 9-year-old, said, "Dad, we were cleaning your room, we found this letter. This guy really knows you." And he described every one of my characteristics and properties that are now, 20 years down the line. I was like, oh my goodness, how could I have forgotten? Let me write to him. I started searching. And then I just realized that Amitabh had gotten into an accident in a bicycle, gotten into coma for the last couple of years. And the week and the day that my daughter told me, he had passed away a couple of days ago. And it just has stayed in my heart for a long time. And I know if he was here, I would want to say thank you, Amitabh, and happy Teachers' Day to him. So I think if you have people in your life that have touched you in ways and forms that have transformed your own journey, talk to them now. Otherwise, it might be too late, and it'll leave this empty heart that we'll never be able to fulfill. And I think many of our mentors that come before us, incredibly shape us, but often enough we are not able to have that conversation. So have that conversation. I'm sorry I have to stop this on a sad note, but I know I'm out of time. Thank you so much for the patience. I'm here for questions. There are lots of tools, so come and enjoy and play with some of these tools.
For opening the universe of life science to us, and for sharing the power of democratizing discovery. Very quickly, we are running late, I'm sorry, but just very quickly, before we go into Q&A, I would like to invite Professor Shashidhara, Mrs. Murty, Dr. Manu Prakash up on stage for a quick felicitation and photograph, and then we'll move into Q&A. Thank you. We are ready for our Q&A session, for which I'll be handing the stage over to Dr. Manu Prakash and Professor Shashidhara. A quick note on a post-event survey. If you did register for the event, you'll receive a survey for after the event on your experience. And the first 100 people to respond will be eligible to win a Foldscope, of course. And then the rest will still be eligible to win a goodie bag from NCBS. I believe it contains some very fancy NCBS branded things. So with that, Professor, Dr. Manu Prakash, please. And the Q&A that was run through the QR code would be closed by now, but your questions would have been collected, and Professor Shashidhara will be asking the questions on stage.
Evening and fantastic talk, Manu. Very exciting, inspiring, and I'm sure many of kids here, many of them are in the school. I think some of them are in the 7th grade. There is some question for a 7th grade student, I guess. Do you want to sit there?
No, we can just talk.
Okay.
This is too grand.
I think Mysore Maharaja and...
two chairs on a stage has a symbolic meaning in India, so... Yes.
Okay, Manu, the first question.
Yes.
You have developed so many tools. From all the tools that you have developed, which one surprised you most? I'm sure you love all of them. You own them. But which one surprised you the most in terms of its impact, or in terms of what inspired you, that some new thing that you discovered using those tools?
Yeah, I think,
Too much light.
One way of thinking about this question is, it's a tough one because it's an iterative process. When you invent something and you share it with the world, you're not the same person anymore. When you make a discovery and you share that with the world, you're not the same person. And since many of these tools happened iteratively, Foldscope has been, we've poured really our most amount of energy and our heart and the longest amount of time. And one thing that it did, it allowed us to think about this idea that the tools are not that important after all. What is important is the community that you build around it. And so for every tool that we build, we first build the community. You know the joke that I had with the Bill Gates tweet, 100,000 Foldscopes for anybody? What I didn't realize at that time, but I realize now, is 100,000 people just exploring and caring and sharing that tool was much more important than the tool itself, because they had the capacity to transform this tool in many different ways. And I think we've now taken that model, so the app store for diagnostics is completely inspired by that, except the community is clinicians. Many of the things that we're doing with local manufacturing of molecular diagnostics is driven by the fact that this will drive, they have the best sets of samples, they have the best understanding. This planktoscope is built out of bamboo because we built this for sailors and fishermen. because we knew that they are the closest to the natural world and an aquatic. I mean, you and I can spend an hour on a boat, and guess what? If you don't take Dramamine, you'll be puking. And here's a fisherman that goes out for sometimes a day or two with a tiny little dinghy, and they are in open sea. I can never imagine doing that. And so I would say Foldscope taught us how to build communities. And now in the class, what I really emphasize is think about the community first. Tools will come, but can you build a large enough community that is passionate, and the ownership of that tool, they feel that they can take it in any different direction. It is hard. It does require resources. You know, somebody actually supported us when we shipped 100,000 of these sets of tools. But one way that I would say is that Foldscope has taught us this lesson, and many other people have now started doing something like that. And it's not something that I learned in a textbook. I didn't learn this in I haven't gone to business school. Maybe they talk about these types of things. But on the other hand, it's been valuable. I mean, I have written some of my most exciting papers by objects that I discovered with a Foldscope. We just discovered the first known example of a single cell that uses origami. So it's a circular thing. We used an origami microscope to discover a life form that uses origami. And now we're using that life form in origami to develop a new kind of a bronchoscope for lung cancer. And so it's like this iterative process of discovery, and it was just in a puddle in a pond, and we collected that data for the first time with a Foldscope, and then when you see something, then you spend 5-6 years trying to understand it. But you know, to be honest, I'm not going to be biased only. It's just every tool is a journey, and you don't really get to decide where it goes. I think this is hard for an inventor to swallow because you build something and just let people play. I mean, it's a little bit like a pencil. People invented a pencil not really thinking how people would use it. You can write poetry, you can write science fiction, you can make a painting.
Great, thank you. Related to tools again, many of your tools are, obviously all of your tools are affordable, accessible, and very simple. And how do you balance the simplicity versus the functionality of your tools?
I think we have this very strict metric we use, cost and performance. In engineering, you know, if you are ever gone through an aerospace program, you have lots of performance metrics, weight metrics. There is a very clear pathway. And you can either build a tool for a few hundred people, or you can build a tool for 1 billion people. And in that scenario, you have to ask yourself a question: do I take a performance hit? And I think on performance, the reason we don't call it frugal engineering, and I absolutely don't call it jugaad, I have to say this explicitly, we call it frugal science because hidden behind these tools was a scientific insight. So for Foldscope, it happens to be a fat length equation. We solve the fat length equation analytically that allows us to precisely make. So sometimes performance is not what you think it is if you apply fundamental science. So when I teach this class called Frugal Science, the title is, we want designers to solve PDEs. There is no applied brain and a basic science brain. It all needs to be mushed in the same person. And so to a certain extent, this is an art. And also, this is also an art of dissatisfaction. We invented a Foldscope. We said we are going to image sub-micron and $1. Simple to explain, has to be below, the resolution has to be below, cost has to be below $1. And then 1,000 ideas don't fit that picture. So until you land on the right idea, you know you have not satisfied your rubric. So set your rubric first, so then you don't cheat. And then just keep going, and it takes sometimes a couple of years to land on an idea that will actually scale.
So another question related to your balancing act. So you've been working in the area which is really high-end deep science, like how cells sense gravity or pressure deep under the sea, or and such kind of questions. At the same time, you're also working on really large societal problems which require citizen science kind of an approach. How do you balance the two? How much time you are sparing for the two?
You don't balance. There is no balance. You try something, you're obsessed about it, and it's very much of a random walk. I think one way that I would say is something critical to me when I started my lab in 2011. I had to put together a website. I was struggling, kind of thinking about, how do I balance the fact that the places I grew up, there were thousands of other kids that were just like me. But why do I get to have the opportunity to have this fancy lab, work with brilliant people? And then I told myself, you balance it with time. 50% of my time had to be dedicated to what I was calling frugal science. The only currency you have is your time. If you care about any of the societal issues you talk about, it doesn't matter what knowledge you have. If you can commit a percentage of time, that's it. And I said 50% has to be that, and that suddenly fills up. And now I struggle because all of those things have grown. But this philosophy has also grown. So I rely on people like Anupma. I'm on borrowed time because her time is then amplifies, and then many people that she has inspired take on that initiative. So suddenly it only scales because behind many of these ideas are, I didn't mention this, for the menstrual pad factory that we just started is a really good friend of mine, Alex. He's dedicated 15 years teaching farmers how to grow sisal. We came up with the idea of sisal can be used to make menstrual pads. He's the one making that action possible because he's training farmers to now build this factory together with him. So I'm relying on him, and again, this is also valuable. If you share, more number of people can engage, and so any large-scale projects cannot be run centrally. There is no central organization that runs Foldscope. Many of you have found Foldscopes and done whatever you have done. I have even no idea. There's no controlling factor associated with it. So I think centralized control doesn't work for distributed projects.
Okay, I think this question must be from a school kid here. What would you recommend a school kid? But in India, by the way, 6th or 7th when they start reading science in a way, and then later in the high school they read more. What does that you recommend to a school kid to see first to get excited in?
Oh,
using one of your tools, I guess.
Yeah, for Foldscope, it's interesting. In that object, there is no recommendation of what you should see. I like giving people a blank piece of paper. And I want you to struggle with it, feel what you should see. The aha moment cannot be fabricated. It's when you're having idli with your parents and suddenly you're like, oh, this is more fluffy, which is a conversation we were having. How much gas is there in this idli versus that idli? Could I understand the size of the bubbles? And I'm making this up right now, but that's what's exciting. It is science, the passion for science is much more internal. It's any subject that you choose is exciting if it comes from you because you feel that this is my discovery. So I think one of the challenges that we have this technical term for it, we call it day science and night science. Day science is written in books, night science is written in nature, and you shouldn't also feel that it has to be a direct, immediate hit. You can carry a tool and not understand its importance for years, and then one day something clicks. So give it time. I think we shouldn't be forcing. This is why sometimes many of the programs, we explicitly tell the teachers that the Foldscope belongs to the kids. How terrible if you give a pencil to a kid, teach them to draw, and then put it back in the library. That's what we do when we share scientific tools. So Foldscope don't belong to the teachers, although they can have their own Foldscope. They should not be locked in the library, and then you will find what you find passionate. I think this is sort of so beautiful about science that, this might sound weird to say, everything is interesting. And that's why I said, if you start observing, you will go insane. And you should be okay with it. Every little thing that you look at has a story, has arrived on this planet, has formed here in some way or form. So I think specialization is for much later. When you've truly found your passion, you will specialize pretty quickly. If you are a young student here, go, look at boogers and scratch your head and whatever you want. There is really no bound to it.
Okay, there are a couple of more personal questions. Looking back your own career journey now, and if you were to be a kid in the smartphone time, what career you would have chosen?
I don't know, I am already trying to figure out what career have I chosen now, right? It's not so clear to me. I have a very hard time what I do. I got an undergrad, I wanted to be an Air Force pilot. I went through the admissions and all of the exams, my dad said, I'm not signing this paper. Do whatever you want, you're not going. I was like, okay, I was super upset. Then I applied for architecture school. I got this phenomenal rank and everything, and then suddenly it's like, oh, why don't we just also try engineering and medical school? It could have been, it's a toss. I started in CS. I kind of felt that I don't like the idea of sitting in front of a computer. I love computers, but I don't like sitting. I want to walk around. That led to kind of a journey in physics. I was like, these are interesting, exciting questions, but look, this is something that we really don't understand. That led me into life sciences. Frugal science was just the outcome of how we all grow up and kind of having empathy. So it's not, now when I think about this, it might feel like a random sort of a path, but we don't tell those stories. So to many of the students who are thinking about careers, I can guarantee you 5 years from now, the world is not going to be the same. So whatever you choose, you probably are not going to be doing the same thing. And it's important to be versatile, and it's important to be honest. Being an honest scientist is very, very hard, because what you really care about, if people around you don't care about, you look like you're working on trivial things. I had lots of colleagues, when I tell them about paper microscopes, they would laugh and like, what the hell are you doing? And I'm okay with it, because I know what I am doing, and so you have to also figure that out, and it can completely change, so you don't feel chained to any field, especially now. It's really, really important to understand that kind of the knowledge dynamics is changing, and unless you can find your passion, it's going to be a very tough battle for you to make truly original discoveries. So it really is a journey. There is no one shot, and I think we should let go. Just natural science is sufficient. We don't really need these terminologies anymore. It was useful in the past, but now we have understood it, and especially for students who are thinking about this, because at the end of the day, you will have to assimilate all of these ideas in your head. They will collide. That will lead to something new. And we know this, and I think some of the programs are doing a much better job. There are many sets of programs, especially for undergraduates, that are trying to build this from very early days. I did apply for a job at Infosys, I remember. I didn't get it. My brother built his career at Infosys. He was there for almost 10 years and then got excited about fiber optics. So I still have his letter, his acceptance letter, and when I didn't get it, I was a little bit jealous. So.
Okay, great.
Yes.
I think in the interest of time, I'll skip the personal question. One last question.
Yes.
Science progresses when observations are made with accuracy and reproducibility in mind. How do you expect the frugal science, particularly citizen science, will ensure that what comes out is trustworthy or reproducible?
Yeah, I think this is something, a lot of people have studied this. There was a phenomenal study done in the Amazon where some scientists had said, can communities and sort of, I don't know the right word, but the tribal communities living in the Amazon, can they really do taxonomy? Can we teach them rigorous taxonomy? And it actually turned out they did much better because they live. It's just that they don't have a language. So the first thing to really understand is, how are you judging what you're doing in a citizen context? Maybe you're using a language that doesn't fit, so that's important. The other aspect of thing is digitization brings a lot of pattern, like for Foldscope, we have digital data sets, we train sets of models, you can look at what the students have discovered, but you can also run ML models, you can also understand many of the other metadatas. We're now building a genomic layer on top of it. So digital technologies bring robustness. So for the diagnostic tools, for example, the user can understand and appreciate and start to find new parasites, while at the same time the base models is helping them find very rigorously what needs to be a clinical diagnosis. And there's a very distinction between what would be the clinical diagnosis, while in research you want to have an open mind. But much of that can be bottled up in a $5 microcontroller at this point. So I think I'm not too worried. I am worried about the opposite, where we are not scaling fast enough. For a question that you care about, or an ecosystem that you care about, if it's disappearing so rapidly, we wake up 10 years from now and I say this in my talks, I am an obituary writer. We just made this discovery in the Arctic. I know in 20 years from now there is going to be no Arctic. So what we just described, this capacity, wonderful capacity of this cell to do this in the Arctic, that's not going to be there. So we just wrote a spectacular obituary. And it feels really depressing in some sense that the state of discovery has to be much faster so you can implement processes to try to preserve it or just even share the importance. So I am completely unfazzled by the fact that, and frankly, a lot of data sets that come out, sometimes we don't even have the right narratives to even understand what is the question. So many community members really ask the right question. We don't ask the right question because we don't belong to that community. We have never actually experienced that problem directly. So I think as scientists, we have to really open up our methods as well. And the community health workers have taught that. There was a huge debate by WHO, should we be investing in community health workers? These people don't have training. Should we allow them to give a medicine? And all of this data has shown that their experience is far more valuable and you can build checks and balances, and reporting is important. But I am a strong believer that I don't think anything else will suffice. You can build data sets and build systems for verification, but I never worry about it.
Great. Thank you very much. It's been a fantastic talk. I think, very rarely see we people who understand the biology, particularly the entire biological world, from microscopic to macroscopic scale. And I think you sort of connected all the living world, which is an emergent property of Darwinian evolution, in one particular continuum. And microbes, obviously, the ones which connect all of these things. And connecting the microbial world to whole of biodiversity and its importance to address the climate change problem and, of course, our health problem is a fantastic evening and a very, very educative and learning experience. Thank you very much. And a big applause to Manu. Over to Neha. Of course, Manu, we can. I invite Professor Raj Ladar, faculty from NCBS, to give vote of thanks.
I'm the faculty at NCBS. I'm also the faculty in charge of a new initiative at NCBS called the Science Education Outreach Office, which I please engage with. I wish to thank the organizers of this talk. So Murty Trust and NCBS, thank you very much for organizing this and all the individuals who did that. Manu, thank you very much for a fantastic talk. Amazing and inspirational. And now I want to thank all of you for coming here. So the talk is aimed at all of you kids to be inspired to become scientists. But it's also aimed at your friends who did not come. Okay? And so please tell them about these opportunities, because the next Nobel Prize winner from India will probably be one of you, will probably be a girl in Nainital who discovers a fantastic carbon sequestration method, a guy in a village in Tamil Nadu who invents a fantastic economic system that causes equality of income. So something like that that solves real world problems is what this inspiration should be giving you. So please go away from here with ignited curiosity, with ignited creativity, but also, as I said yesterday, with courage and conviction that you can make a change. Okay, so thank you all for being here. Thank you very much, and thank you, the organizers. We'll take care of that. Okay, so thank you all.
I said a big thanks to NCBS for pulling off this fantastic event. I'd like a show of hands from all the volunteers who've been working very hard since 2:30 p.m. today. Please do take a bow. A special heartfelt thanks to my colleague Manal, who has been the backbone behind this. She's right here. It would not have been possible without her. So now, friends, please join us outside for refreshments. My colleagues will be announcing shuttle departures to the Majestic Metro Station shortly. So get talking about Foldscopes and what you would do with them, and enjoy some snacks and some tea and coffee. And again, thank you very, very much for taking out the time and being here today and sharing this experience with us. It would not be possible without you here. Thank you.